Use my audio here.
I think I had a very good kind of idea about your backgrounds. So my today's talk is gonna go through really basic and simple principles in the field, and I will try to end up with the facilitation with some of my own research on the delivery of messenger RNA for new therapeutic applications.
But before we go through some of the real science talks, I'll just gonna give you an introduction about myself. So my name is Saed. I'm originally a pharmacist. I was born in Jordan, and then I finished my undergrad degree, and I wanted to do my PhD abroad. And I found out this Japanese scholarship that they gave me full tuition fees, full stipend, and applied. And I spent 8 years and a half in Japan. I did my master's and PhD, and I also did a little bit of postdoc there.
A little closer, maybe, right?
Mhm,
Yeah.
right. So
I have to say.
yeah, I'm gonna do it like that. And then I moved to the United States. I continued my postdoctoral research in Hopkins. Similar topics, but different therapeutic applications. So what we do is nanomedicine, and we just apply it to different disease targets. And actually, just like 2 days ago, I signed my contract at Hopkins as a non-tenure track faculty position. So that's gonna be some exciting new chapter in my life, where it's not ideal. It's not a tenure track, but we know how difficult nowadays getting a faculty position is. But I'm very excited because I can write grants and kind of further move on with my research program. So for today's goals, I just wanted everyone to know a little bit more about what is the future of medicine and why nano is important. So when we talk about the future of medicine, we'll always need to talk about drug delivery, because a drug that is not delivered is not a drug anymore. Because drug delivery nowadays is as important as drug discovery, let's say 10 or 20 years ago. And also I want to open like a dialogue if there are some misconceptions about gene therapies, messenger RNA, mRNA vaccines, nanotechnology, and see what you guys think, and like we can discuss different topics. And I would like that to happen as an open dialogue. So if you have any comment or any question during my talk, please interrupt me and ask. So if we really think what scares us the most in life, what would it be? What scares you most in life?
Dying.
Yeah. That actually is probably a relief. Because for me personally, I am really scared of disease or suffering, because there's something in us that we don't want to suffer. We have really low tolerance to pain or suffering in general. And I think all of our human thinking and innovation has been always centered around how we can reduce suffering and how we can increase the quality of life at the same time. But not every disease is actually suffering. There are like simple diseases, and there are really more complicated or intractable diseases that are not easily treated. So for example, what do you do when we get the flu? We'll probably just suffer. Maybe I should end it with the flu, because it's the most sufferable disease, right? So we probably just gonna take some painkillers. If you have a fever, you're just gonna take some Tylenol. And if it was just a common cold, that will just resolve in 2-3 days. And if it was the influenza, you probably have to stay in bed for a week and take meds and reduce your fever until you're recovered. And what happens if you have seasonal allergies or some sorts of like eczema or some sort of any kind of immune reaction to allergens? You will probably just take some antihistamines. And these antihistamines,
they will just like stop your itch or stop your cough or like dry eye or itchy eyes. And that depends on how bad and how long this allergy is gonna last. Your also treatment gonna last for that amount of time. But roughly we're talking about a month to 3 months, depending on what kind of seasonal allergy you have. So how about diabetes? So there are multiple types of diabetes. Some people are diagnosed with diabetes when they are kids, or some people they are diagnosed with diabetes when they are adults. But once you get the diagnosis, you'll probably be prescribed like a glucose lowering medications, or you can get insulin itself depending on how severe your diabetes. But most probably, once you get the diagnosis, you're gonna take these drugs lifelong. So if we look at all of these examples, from flu to cold to seasonal allergies, or even diabetes, what is the common theme between all of these previous cases? Let's all think together. No wrong answers. What is common between these diseases?
You're treating and not curing it.
Yes, that is correct. You're just doing symptomatic treatment. And symptomatic treatment is actually when you treat the symptoms or the signs of the disease without tackling the origin or the root cause of the disease. So it is literally if you have a smelly trash can in your house, and instead of tying up the bag and throwing it away, you just like spray some air refreshener and just live up with the situation until you smell the bad smell again and spray it one more time until you finally reach a point to reach a conclusion that you actually have to throw away the garbage. So if we have to choose one of these, if you are asked to choose one of these in your life, to be sick and have to visit the hospital every week, or visit the hospital every month, or every year, or never need to visit the hospital in your lifetime, what would be your choice? I mean, I would be surprised if anyone picked any other option other than 4. And that's because we just hate diseases, and the less we need to go to the hospital is better for us. And that's why we need some treatment that is not symptomatic anymore. We need to actually deal with the root causes of diseases. And as Buddha teaches, so to be able to know what is or how to solve suffering in life, you need to find what is the origin of this suffering. All the Buddhism teaches that suffering comes from desire or wanting things, but biologically speaking, what is the origin of diseases? And anytime we have to think of the origin of diseases, we always have to go back to the central dogma of biology. The central dogma of biology, although this is a very simplified version of it nowadays, it entails that every cell in our bodies holds the whole genetic makeup for every single function in our body. And this genetic information is held in the DNA. And because this DNA is very precious, stays inside the nucleus, and it communicates with the rest of the cell through a messenger. And this messenger is known as messenger RNA. So it holds a copy of the precious DNA and brings it outside of the nucleus. And then the mRNA is translated to a protein. And protein is actually the building block of our bodies because it is about the 55% of the dry cell mass, and it literally
protein, like any functional molecule in our bodies, whether it was a cell receptor, it was an enzyme, it was an antibody, or it was a transporter, is a protein. So almost every disease we know about comes from a dysfunction in a protein. Whether there's too much protein in the cell, or there's too little protein, or not enough or insufficient amount of protein, or that actually we have the protein, but we have a faulty version of it or a mutated version. So for example, in some cases of cancer, and in this case is I'm just gonna try to, okay. Yeah, so you can see the head of it. Some types of cancer we have, and in this case, we're talking about some sorts of breast cancer. There's a cell receptor, which is a molecule or a signal like an antenna expressed on the surface of these cells called HER2 receptor. And once it's overexpressed or upregulated in number, it can bring in a cell transduction or proliferation signal, which makes the cell divide in an uncontrolled manner, giving rise to a cancer or a tumor-like situation. And that is a situation where too much protein, such as the HER2 receptor, causes disease. In other cases where, for example, in arthritis, when we have proteins that are responsible for making cartilage, which is kind of like the cushion that lines the joints and protects them from friction. So when there's like cartilage-making proteins are downregulated, or we don't have enough of these proteins, we're gonna develop a disease situation such as arthritis, or painful when moving, like a painful situation when you're moving your joints, knees, or elbows, or any other joint in your body. Other cases where actually we have the protein, for example, hemoglobin, which is a protein available in our cell bodies and responsible for transporting oxygen to the rest of our body, is usually has a spherical or like a globule shape inside the cell. But sometimes there's a genetic mutation that causes a deformation in its shape. And once there's the deformation, it will have a negative implication on how the cell functions. And in this case, a patient will develop a condition called sickle cell anemia. So what in my field, we studied a group of drugs, what we call genomic medicines. And genomic medicines is just an umbrella term to define or include any drug that can be used to interfere with one or more steps of the central dogma of biology. So let's see some examples of what are these genomic medicines, that the final goal or the intention is actually to treat the disease by its root cause, so that we are not reliant on symptomatic treatment only anymore, but actually correct diseases and end suffering, and we never need to go to the hospital again. So for example, some of the genomic medicines that can be used to correct diseases that originate from too much protein is that actually, for example, the mRNA is usually translated to protein, but if we have too much protein, how about if we silence this mRNA? There are specific molecules known as siRNA, or small interfering RNA, or microRNA, that can be specifically guided and target this specific mRNA, and then they degrade. And once the mRNA degraded, then there is not enough protein, or we're gonna lower down the amount of this disease-causing protein. These kind of molecules, the siRNA or microRNA or ASO, can be used to reduce protein production by interfering with the messenger RNA. But we can also reduce protein production on the DNA level by inducing genomic modifications. For example, using CRISPR-Cas9, which is a new technology that is literally described as a molecular scissors that can induce target cleavage in the DNA. And once you disrupt this gene that encodes for this specific protein, you're gonna reduce or lower the amount of this disease-causing protein in the cell. So how about if we have a disease that originates from too little protein? So what we need is simply to supplement the cell with the protein that it doesn't have enough amount of it. So in this case, we just need to supplement with, for example, additional mRNA. So we introduce the cells with messenger RNA. And once the messenger RNA inside the cell, it will use the cell machinery that is already existing in the cell, such as ribosomes and other enzymes, and it will produce the protein by itself. Or we can also introduce to the cell a DNA segment, which also encodes that specific protein, and it will produce the mRNA that has a copy that holds information of how to make the protein, and then the mRNA will make the protein itself. So, and again, like in a situation where we have the protein, it's not a problem of having too much or too little, but the type or the quality of the protein is not good enough. Such as in cases of cystic fibrosis or sickle cell anemia. And the field has been revolutionized in a way that we can actually induce very precise genome editing by correcting or replacing this faulty gene and replacing it by the correct DNA sequence that encodes for the right amount or the right type of protein. So, and one of these like most advanced technologies used to induce or to introduce genomic corrections is CRISPR-Cas9. So CRISPR-Cas9 is a disruptive technology that won the Nobel Prize, I think, in 2020. And so the field of genome editing is more than 20 years old. But what this Cas9, CRISPR-Cas9 brought to the table is that now we can induce genome editing at a much higher rate and with much higher specificity with lower off-target modifications. So it's literally like we have a scissors that induces or introduces a cut in the DNA, but this scissor is blind, and to guide it, we use a guide RNA, which is literally like a GPS that tells the scissors where to go exactly on the DNA. And once these two systems, the guide RNA and the Cas9 scissors or the enzyme, then they will go to this DNA, induce the cut in the DNA, and then we can paste the or remove the faulty segment of the DNA and replace it with the correct version of the DNA. So we talked a lot about different modalities or different ways of how we can induce precise genomic interventions, but actually the problem is delivery. So delivery is the problem. Because when we look at a typical genomic drug or genomic medicine, we've heard a lot about siRNA, microRNA, mRNA, guide RNA. So most of these drugs are actually nucleic acids. And a nucleic acid is this molecule that is composed of phosphate groups, ribose, which is just a sugar, and the common CGAU that they hold the genetic information in our bodies. So this nucleic acid as a molecule is actually very big. So if we talk about a typical, for example, let's focus on messenger RNA as an example of these nucleic acids. And one molecule of mRNA is almost 4,000 times larger than a glucose molecule. So they are actually very large and they are extended because it's like a polymer. So because they are large and they are highly negatively charged because of their phosphate groups, they cannot enter the cells because our cells are negatively charged because we have a high amount of glycoproteins that cover the surface. So negative and negative, they repel each other, and then the mRNA will not be able to enter inside the cell anymore. And the mRNA is also very not stable because if you remember when we talked about the central dogma, mRNA is typically inside our bodies, but inside the cell. So if we have mRNA outside the cell, the body is going to sense it as a danger molecule and is going to chop it off because naturally, mRNA or nucleic acids should not be outside the cell. So in my field, nanomedicine is dedicated and focused on how to smuggle these nucleic acids specifically to inside the cell. So if you consider, or if you look at the nucleic acid drug as a fragile vase, and if you want to ship it to a different location, you always have to start with wrapping it with a bubble wrap. So that's protection. And that's not enough because you need also to label it with a shipping label and send it to the right address before somebody leaves it on a doorstep that is not the intended delivery location. And this is what we exactly do. We formulate these nucleic acids. So here in this image, nucleic acid is this coiled strand inside. Inside, very tiny, tiny capsules in the nanoscale to protect them from degradation. So if they are inside these tiny envelopes, these RNases will not penetrate, and therefore the mRNA will stay intact. And because now they are covered, the negative charge we talked about is going to be masked. And therefore, the immune cells will evade the recognition. And then the target cell, is going to be able to take this envelope much easier. And what we use, so there are actually many, many different ways where we can make nanoparticles. We can use polymers, such as nanoplastics, or we can use lipids and fats, which are these bouncy kind of little molecules that are natural and also present in our cells. So cell membranes or lipids are the barrier that protects the cell from the outside microenvironment. So what we can is we can extract these lipids from animals and plants and make these nanocapsules and put the RNA or the nucleic acids inside them. And the most clinically advanced lipid nanoparticulate system for the delivery of messenger RNA, and most probably you already have it in your left or right arm if you have taken the COVID-19 vaccine, is something we call lipid nanoparticle. So a lipid nanoparticle is composed of 4 types of lipids, what we call a helper lipid, ionizable lipid. Ionizable lipid is a positively charged lipid. So if we remember, we mentioned that the mRNA is negatively charged, and we complex it with the positively charged lipid, and by doing so, these 2 molecules will self-assemble into this nanostructure. And we also need other helper lipids such as cholesterol, and cholesterol is also present in our cells to give it fluidity and reduce its rigidity. And also we have other polymer or plastics that give stability to these, the nanoparticle in a molecule called polyethylene glycol. Polyethylene glycol is commonly used in cosmetics and shampoos and any other formulation on shelf in pharmacies. And it gives this colloidal stability, meaning that the particles cannot coalesce or cannot kind of fuse with themselves, and they stay suspended in a solution for an extended period of time. So we talked about nano a lot, but what is actually a nano? What is nano in real life? So I'm going to show you a small segment of a video that we I made in Japan as part of science communication activities and for kids. So it's going to be in Japanese, but there are subtitles. Only if I know how to. Okay. Wait. I think I'm. Why can't? Why is the? I'm trying to. Is there a volume or? I have no idea how to.
It's okay, it's in Japanese anyway, right?
Yeah,
Maybe
so that
just as fine
just look
as
at
to read
look
it.
at the subtitles then. Okay. I'm actually very happy that there was no any sound because I hate hearing myself again in this video. So actually, I was talking with a nanoparticle. Of course, it's Japan, so then it needs to be an anime somewhere. So we were like in a journey with this nanoparticle. We were like shrinking to a nano size. We were traveling inside the human body. And yeah, if you want, you can scan the QR code and watch the full video. And in that video, we actually described about how we can use nanomedicine for cancer treatment. So again, nanomedicine is very useful and very versatile. It's not only for delivering genomic medicine, but also can be used to deliver any type of drug. And the reason why we need nanomedicine, especially in the context of cancer, is just because cancer drugs are very toxic. So when we deliver a dose of cancer drug, only a very tiny fraction of that, below 1%, goes to the tumor tissue, and the remaining of the drug goes to your hair follicles. And that's why people like start to lose their hair. Or it goes to your immune cells, and that's why you have a weakened immune system, and you become more susceptible to infections. And when we put these cancer drugs inside nanoparticles, and we put a shipping label that is specific to tumor cells, we can specifically target them to cancer cells and avoid other types of cells. So circling back to the mRNA, and COVID-19 vaccines was the biggest real life trial or a clinical trial to actually test the efficacy of these non-viral vectors versus viral vectors. And the evidence is huge that mRNA vaccines are actually very effective in reducing the severity and deaths related to COVID-19 vaccine. So COVID-19 vaccine, whether it was the Moderna vaccine or the Pfizer BioNTech vaccine, they were composed of these lipid nanoparticles that we just discussed about. And they have an mRNA that encodes for a fraction or a small part of the COVID-19, which is the spike protein. These lipid nanoparticles are injected in the muscle. And once they are inside the muscle, they are taken up by either the muscle cells or the immune cells in the muscle. And once they are inside, then the cell will recognize it as mRNA because our cells are able to read mRNA freely because mRNA is in every cell. So they will read this transcript and then they will translate it to a spike protein. And then the cells will present this spike protein on the surface. And then this is what drives the recognition of our innate immune system or innate immune cells with these cells that they express the spike protein in the surface, and that what will cause the immune recognition and production of cellular immunity or antibody immunity to fight the real infection when it happens. So mRNA has also been studied for 30, maybe, let's say 20, 30 years. And also lipid nanoparticles were also being studied in labs for 20, 30 years. And what happens during the pandemic is that these two technologies merged together and they were very effective in mass production of vaccines in a very, very unprecedented way. So last actually week, I was in Boston for a conference and I met Kathleen Carico and Drew Weissman, who are the Nobel Prize winners for their important discoveries and pioneering work on mRNA vaccines. So the nanomedicine field was a little bit disappointed because we also wanted someone from the nanomedicine to also win this prize, but hopefully in the upcoming years. But again, that the mRNA and the LNP work has been separate, and they united during this pandemic. Oh, she actually signed. Kathleen Carico has a very nice book. It's called the Breaking Through My Life in Science. She has a very inspiring story. She's Hungarian, and she was raised and born in Hungary, and she moved to the United States to pursue a scientific career. And she was so poor so that she has to sell her car to afford the ticket and primary accommodation when she first moved in. She initially worked at the University of Pennsylvania, and because she was working on something that is so strange at that time called mRNA and nobody is interested in, she was actually demoted, and because she was not productive enough, and she was unable to secure as many NIH grants as other her fellow faculty at UPenn. And then I think she left University of Pennsylvania, and that's why she worked for BioNTech in Germany. And, but yeah, she won the Nobel Prize just a year ago. So the LNP technology that was used in the mRNA vaccines was not new. And as I mentioned, there's like research on developing lipid nanoparticles for 20 or 30 years. And the first clinical example of using lipid nanoparticles in human bodies was actually approved in 2018 to deliver siRNA, or small interfering RNA, which is a molecule that targets a specific sequence in the innate mRNA in our cells. And once it inhibits it, it reduces the production of a protein. And in this case, that specific drug called Onpattro was targeted to silence a protein or mRNA that encodes a protein called transthyretin. And the way it works that these LNPs that they have the siRNA inside them, they are injected in the bloodstream. And once they are injected in the bloodstream, they will encounter so many different proteins in the body. And one of these proteins that has specific binding or preferential binding to these LNPs is called ApoE. And once this ApoE binds to this lipid nanoparticle, it will be taken up by the liver because now this kind of like a protein corona or the protein absorption on the surface of the LMP makes it very desirable for taken up by liver cells. And once inside in the liver cells, there's also a multiple steps of engineering these LMPs so that this siRNA can escape the degrading compartments known as the endosome or the lysosome. And once it escapes that acidic or degrading compartment, it goes to the cytosol, then it will find its mRNA. And once it binds to this mRNA, it will degrade it, and the end result will be reduction in the amount of a protein that we don't need much of it. So the clinical trial was very significant, and it was one of the first examples where we can actually start to dream with more confidence about making drugs that induce corrections with so many little interventions. Because in this study, they did only one dose of siRNA, and you can see that the reduction in the this transthyretin protein was sustained for up to 1 month. So this disease, they didn't have any drug effective for it in the market or in the clinic. And this new drug is not only effective, but we don't need much of it. So once every month is, I think, was choice number B in that question. But still, now we are working, like scientists working on genome editing technologies where you can actually correct the gene forever. And there are actually clinical trials where you can use an injection once every year. So we are moving from situations where there are no approved drugs for that disease to very effective treatments that are needed once every month to a newer treatments that are needed once every year. So I think the future looks very bright and very promising.